We're going to be talking about Bengston research today. I'm not going to give you any type of review. All that I will tell you is what happened, and we're going to talk about the facts. We're going to read about some on the website. We're going to read about a little bit on a research paper and just go through it one by one. This is a felt tip pen. It is my most favorite pen in the whole wide world. However, up until a couple of months ago, I had not had a felt tip pen in years, probably 8 or 10 years. Going through the Bengston research course, one of the things they talk about is image cycling. In image cycling, you are supposed to do a certain thing, and when you do that thing, the things that are on that list are supposed to come to pass. Now, this felt tip pen was on my list. It was like number 3 or so, and lo and behold, my wife presented this to me. She had bought it on Amazon, and I asked her, why did you buy this felt tip pen for me? And she said, oh, I don't know, I just thought you would like it. And then I asked her when she bought it, and lo and behold, she thought about it around the same time, about a day after I started doing my image cycling. That's a fact. That's what happened. But what does that have to do with healing research? Well, we'll get into it, ask your questions later. But let's just start reading. I'm going to click on the healing method. The method. The Bengston Energy Healing Method is an innovative healing technique that has been shown to be effective on a wide variety of conditions. The core of the Bengston method is image cycling. That's what we talked about just a minute ago. Details about the method and how it evolved can be found in the book The Energy Cure or the 6 audio CD set Hands-on Healing. Okay, so this is what he's talking about, the 6 CD set. It's also in audio downloads. Each one has, you just go through and you listen to it, and it explains everything perfectly. And it looks like this. It's only like $39, so it's very affordable. Anyways, where were we? In the 6 audio CD set, Hands-on Healing, a training course in the Energy Cure. The technique is largely mechanical and belief-free, but like any new skill, it takes practice and commitment to become proficient. The applications of my techniques have been and continue to be tested under strict laboratory conditions in many independent labs and medical schools. Many peer-reviewed articles based on these evaluations have been published in professional biology and medical journals. This does not make my data true, but it does give the added benefit of having gone through rigorous scrutiny. The bottom line take-home message from all of my techniques is that you should first and foremost have fun. A playful approach to an area so little understood will take you a long way, and once you start having success, it is equally important to avoid ritualizing any technique. Freshness matters. Ritual is just for safety. I assume that the methods will evolve and improve. Now, and this is Dr. Bengston. Just a quick note, what you see him doing, there are mice in this little jar right here. He is sitting here with his hands on here doing some image cycling. That's what he's doing. What is image cycling? Image cycling, for reasons I do not yet understand, I don't think anybody does, image cycling, for reasons I do not yet understand, is extraordinarily important in enhancing healing. The cycling technique involves making around 20 mental images of things you would like. All of my techniques are selfishly based, and I will put they are selfishly based. One of mine was that felt tip pen. Or situations that you would like to be in the future. The mental images cannot be general, like I want to be happy or I want to be healthy. Instead, the images must be able to recognize when accomplished. They must be able to be recognized when accomplished. For example, here I am driving my new car, or here I am in some specific exotic place. With practice, which includes, in all likelihood, a refinement of the images through a number of lists, these images become more specific and idiosyncratic as you find out what you really want. The image cycling itself involves running through these images at very, very fast pace while you are otherwise engaged in ordinary activity. Like any skill, at first this is awkward. Think learning to ride a bicycle. After sufficient practice, riding a bicycle becomes effortless and second nature. What seemed like a formidable task requiring focused attention, riding a bicycle, I clicked off of it. What seemed like a formidable task requiring focused concentration, like riding a bicycle, can now be done mindlessly. Image cycling is like that. In our experience, image cycling seems more difficult to learn on one's own. That's a reason for the workshops and the audio training set. Now, if you look right here, you will see some mice. See the tumors? I'm going to blow this up a little bit. Healing is not linear. In the mice research, there is a sudden transition between day 22 and day 26, or 6 days. The tumor in the mouse above completely healed. In the center photo, right here in the center photo, the fur is being pulled back. It's very difficult to read so small. The fur is being pulled back to find where the tumor was. At day 35, the mouse continues to be healthy with no indication of cancer. And by the way, before I go any further, this it had like an unbelievably high success rate, and you can learn about that later. Research. In the 35 plus years that I've been doing research on healing with my method, I can't tell you the number of times that the tests of my ideas under controlled conditions results in me being totally surprised. What I thought I would find does not match my actual results. I have learned not only patience, but most of all, humility as I ponder the mysteries of healing. I can tell you without reservation that no one, including me, has a lock on the truth about healing. My previous beliefs about healing have repeatedly been shattered by the data that comes out of the lab. And this is a wonderful thing. It actually turns out to be quite liberating to do research, as the wonders of nature are past my feeble imagination. And, as far as I know, the imagination of anyone else. I have found too many believers convinced of the truth of what they think they know, and I suspect under controlled conditions much of the truth of what is out there would not stand the test of serious scrutiny. And so be cautious, be skeptical about everything, even what I say. Let me say that image cycling is not a healing technique. It can be used for healing, and in the workshop we discuss hands-on healing techniques for combining cycling with healing. However, it is a practice that I recommend whether you want to heal or not. There are many positive experiences reported by people who cycle, in addition to the interesting phenomenon that the things on the list seem to become reality. That's exactly what happened to me on more than one occasion. So I have looked at the brains of image cycling users, EEGs, fMRIs, and something different is going on here. See papers, whatever papers are. We have found unique harmonic frequencies in the brains of practicing cyclers. In addition, simultaneously running tests on both healer and healee demonstrate that something akin to brain phase locking between the two occurs. Once again, we do not know what this means, only that something is clearly going on that appears to be unique. I continue to do the research. The scroll of questions and interest is long and perhaps endless. And about Bill Bengston. Excuse me. Before I even read this, I'll tell you that the first interview that I saw on YouTube about Bill Bengston, I thought he was kind of a dork. I mean, he came on there and he said, "Oh, I've been doing this research for 140 years and blah, 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 blah." And I was like, do you honestly think you're 140 years old? Now, of course, he was joking at the time, but I mean, there's a bunch of whack jobs out here, so you just don't know. No, that's just a little joke. So if you see him talking about how many years he's done this, 140 years, 200 years, whatever, 90 years. It's just a little joke there. Okay, I love his work. Anyhow, William F. Bengston, or Bill, is a professor of sociology at St. Joseph's College in New York. He received his PhD from Fordham University in New York in 1980, and his day job areas of specialization include research methods and statistics. Bill has conducted research into anomalous healing for over 35 years and has proven the effectiveness of his technique in 10 controlled animal experiments. Actually, if I'm not mistaken, I think that's more. I don't know. Do your own research. Conducted in 7 university biological and medical laboratories. His healing research has produced the first successful full cures of transplanted mammary cancer and, here we go, methylcolin something induced sarcomas, which is a cancer, in experimental mice by laying on of hand techniques that he helped develop. So, I'll just continue reading here. He has also investigated assorted correlates to healing, such as geomagnetic micropulsations and EEG harmonics and entrainment. Dr. Bengston does not provide healing treatments, medical diagnosis, or any other personal consultations related to health, medical, or psychiatric issues. Rather, his time and attention are devoted to energy healing, research, and education. Dr. Bengston has publications in the Journal of Scientific Exploration, the Journal of Alternative and Complementary Medicine, and Explore. Where are they? They're up here somewhere. I'll show those to you. In addition, he has lectured widely throughout the United States and Europe. Bill has been a member of the Society for Scientific Exploration since 1999 and currently serves as president of the organization. He also is on the editorial board of the Journal of Alternative and Complementary Medicine. Bill has written a memoir with Sylvia Fraser about his healing experiences and research entitled The Energy Cure, Unraveling the Mystery of Hands-on Healing. A CD audio instruction program, Hands-on Healing, a training course in the energy cure is also available from Sounds True, which is, where's it at? Right here, up here where it says publications. That's what they're talking about here, the book and the course. And some of the articles are listed on the site as well. And you can go to the site, which will be listed in the show notes below, and research all of that. Here is a bunch of videos on YouTube. It's all on the site. Go check those out. He's really interesting to listen to. Here are the journals listed above: the Journal of Scientific Exploration, the Journal of Science and Healing, the Journal of Alternative and Complementary Medicine, and Edge Science. Also, there's a forum if you want to go to. I mean, you have to ask for it, but whenever you go to the forum, there's a lot of people talking about, well, disease conditions, experiments, healing experiences, charging, cotton, water, etc., and things like that. So let's switch gears and go over to the paper. This paper was put out by the U.S. National Library of Medicine, National Institutes of Health, through the NCBI. The name of it is Transcriptional Changes in Cancer Cells Induced by Exposure to a Healing Method. I have not read this paper yet, but I do know some of the names are Sarah Bissami, I don't know how to say the last name, William Bengston, which that's the guy we just talked about, Dean Radin, which is he's over at the Institute of Noetic Sciences. He puts out a lot of good stuff. Look him up on YouTube. Michael Turner, not sure who he is, and John McMichael of Beech Tree Labs. Okay, so let's just get into it and see what the research and data says. I will not read it word for word, but I'll read most of it. But sometimes when it gets into some of the things, like I don't know what a QRT PCR is. Don't know. And I'll probably just skip over that. Abstract. Energy, and mind you, this is a medical, journal type area. It's not just a ho-hum, woo-woo, this is an actual serious thing. It's the same exact place they post research for everything else, for any medicines that you might take. Okay, here we go. Abstract. Energy healing, or healing with intent, is a complementary and alternative medicine therapy reported to be beneficial with a wide variety of conditions. We are developing a delivery technology for a method previously tested in mouse models with solid tumors, the Bengston method, independent of the presence of a healer. The goal of this study was to assess whether stored or recorded energy has an impact on breast cancer cells in vitro using energy-charged cotton and electromagnetic recording of healers practicing the method. Electromagnetic recording, like an audio recording of the healers practicing the method. Expression of genes involved in cancer and inflammation pathways was measured by quantitative reverse transcription polymerase chain reaction, or this thing right here. I guess that thing that I didn't know, that's what that stands for. Treatment of cells using energy-charged cotton resulted in statistically significant changes less than 1.5-fold. In cells exposed to an electromagnetic recording, 37 genes of 167 tested showed a greater than 1.5-fold change when compared to the control. Arid 68 genes showing statistically significant fold changes. 2 genes, ATP citrate lyase, I don't know, and interleukin, I don't know, were consistently downgraded at 4 and 24 hours of exposure to the recording, respectively, in 3 independent experiments. Okay, when it gets into this right here, I'm not sure. Let's just see what we can derive more. I'll finish reading though. Both ACLY and IL-1B were also downregulated in cells. Downregulated, I understand that. Your genes can be downregulated in cells exposed to hands-on delivery of the method, suggesting that these 2 genes as potential markers of the healing method. Man, they should just call me the research butcher, because I just butchered that. Whatever, let's continue. Introduction. Virtually all recorded societies report that certain individuals appear to have the ability to heal. Oftentimes this healing has been associated with spiritual disciplines of one sort or the other, and the healers themselves have sometimes been accorded a special status within the culture. Healers have utilized various methods of practice, including laying on of hands, prayer, and induced altered states of consciousness, to name a few. The father of Western medicine, Hippocrates, referred to this healing as the force which flows. Lost my place here. Where are we? The father of Western medicine, Hippocrates, referred to this healing as the force which flows from many people's hands. Historically, the source and mechanism of the healing evaded systematic research, even as the number of clinical cases greatly increased. The foundation of modern Western healing research can be traced to the pioneering work of the late biologist Bernard Grad at McGill University. I'm going to stop right there, and I'm going to copy Bernard Grad because I've heard this name many, many times, and we're going to research him at a later date. I'm just putting Bernard Grad in here so that I can, there we go. I will do that later. In carefully controlled experiments, Grad found that selected healers could influence the germination of plant seeds, the growth rate of plants, and the curing of seeds that had been shocked by saline solution. In addition, he was able to measure the ability of healers to reduce goiter and stimulate wound healing in mice. Since Grad's initial work, there have been innumerable preclinical studies of healing, sometimes categorized by the target of the intended healing. Benoit, I don't know, whatever. For example, discusses healing action on enzymes, cells in the laboratory, fungi, yeast, bacteria, plants, single-celled organisms, and animals that have been subjected to a controlled study. And I'm going to copy this one too, because you know what? There we go. The proliferation of healing studies has continued to rise in recent years. At present, there are several peer-reviewed journals devoted exclusively to the burgeoning effect of complementary and alternative medicine, publishing both preclinical and controlled clinical studies of healing of a wide variety of conditions. In addition, there is an increasing number of peer-reviewed journals which are not focused exclusively on CAM or complementary and alternative medicine, but they are open to publishing controlled studies in these areas. Presently, there are a wide variety of healing methods in active practice, the most well-known probably being Reiki. There is virtually nothing known about how the various methods of healing converge or diverge in terms of healing efficacy, nor are there data which can distinguish whether there are differing mechanisms of action utilized by the different healing methods. Anecdotally, Reiki, for example, is thought to be, you know what, I'm going to have to look up how to pronounce this word. If this word is efficacy, then this word might be efficacious, and that's the way I'm going to pronounce it. So, anecdotally, Reiki, for example, is thought to be efficacious, so let's just look up that word and see what it means. Of something inanimate or abstract, successful in producing a desired or intended result, effective. So, efficacy, there we go. The ability to produce a desired or intended result. So if something has efficacy or is efficacious, so how do you say it? Efficacious. Yep, efficacious. So let's read that again. There is virtually nothing known about how the various methods of healing converge or diverge in terms of healing efficacy or in terms of healing success, pretty much. Anecdotally, Reiki, for example, is thought to be efficacious for pain and anxiety, or it can work for pain and anxiety, but not for conditions such as cancer. The method utilized in the present study seems to be somewhat unique in that it has a history of apparent efficaciousness or apparent workingness with some cancers as well as a variety of other disorders. The Bengston energy healing method. The term energy healing, let's see if this says anything different than what we just read earlier on his website. One second. Yeah, there's some other stuff. It's pretty cool. I'll read it. The term energy healing is somewhat presumptuous in that no energy has been isolated in any healing modality. The use of the term energy in the Bengston method is for practical reasons, in order to conform with the common parlance, or the common usage. Like what they're talking about is he talks about energy healing, but it's actually maybe not energy. We just don't know. The method came about 45 years ago when Bengston began to observe a New York-based healer treating a wide variety of conditions in a clinical setting and to investigate the possibility of developing specific healing techniques which might become reproducible by others. After years of exploration, a method of very rapid imaging was developed that seemed to be efficacious in others, producing the patterns produced by the original healer. And what that means is the imaging that they're talking about is the image cycling. Detailed descriptions of these rapid imaging techniques have been described elsewhere in the hands-on healing course right there. Among the more interesting observations with the New York-based healer, as well as those who practiced the rapid imaging healing technique, was clinical success in malignant growths and lack of success with benign growths. So they had success in malignant growths and a lack of success in benign growths. What I'm going to assume is they had success in growths that were growing really fast, but not so much in growths that were growing really slowly, like maybe a wart or something. Equally interesting, the healing could apparently be transferred and stored in materials and applied later without the healer present. These patterns were similar to those found experimentally by Grad, with cotton, water, and crystals apparently being especially receptive to healing storage. Now that's really cool. Cotton, water, and crystals. Okay, let's continue reading. In the mid to late 1970s, Bengston began looking to follow Grad's experimental approach to healing and moved into a systematic experimental work with animals. With David Crensley and Marvin Wasserman at Queens College of New York, Bengston started to investigate the effects of healing techniques on biological models widely used in conventional research. The synergenic model of C3H/HEJ mice I don't know what that means. Mice engrafted with mammary carcinoma cell line H2712 was used, which normally results in a 100% fatality of the recipient mice within 27 days. And that means all of the mice died within 27 days, 100% of the time. These in vivo experiments involved inexperienced and skeptical faculty and student volunteers trained for 6 weeks in the Bengston method, placing their hands around mice cages for approximately 1 hour a day while practicing the rapid imaging techniques. Instead of the predicted fatality within 27 days, they found reliable full lifespan cures of the mice. And you can see here and here there's a couple of notations. Engrafted mice developed tumors within 14 days, as expected. Around day 21, tumors in treated mice started to ulcerate, and between day 28 and day 35, tumors resorbed until full remission. That's cool. That means that some of the mice were healed, I guess. Because of the unconventional nature of the research, Bengston insisted on independent replication by disinterested researchers in independent labs. As of this writing, more than a dozen healing experiments using the Bengston energy healing method on this cancerous mouse model have been conducted in 6 independent labs. Subsequent to the determination that the healing effect was reliable, secondary correlates involving the effect of distance, dose, and whether healing can be stored were introduced. In brief, healing does not seem to diminish with distance. There is minimum dose necessary to produce the healing effect, and treated water and cotton seem to be able to reproduce cancer cures without further human intervention. Now, we're looking at NCBI, an NIH government thing, and it's talking about water and cotton seem to be able to reproduce cancer cures. Let that sink in for a minute. Studies of the biological effects of the technique on human healers and healees have been conducted to investigate its effects on brain activity by the electroencephalogram, or EEG, and functional magnetic resonance imaging. Those studies indicate reliable physiological changes in both healer and healee. Paired recordings of the healer and subject, done both with the subject at a distance and in proximity to the healer, showed harmonic frequency coupling across the spectra, followed by between individual EEG frequency entrainment effects, and then by instantaneous EEG phase locking. These results suggest the presence of a connection between the healer and the healee, and may thus provide a mechanism for phase coupling. The connection seems to be stimulated by the needs of the healee more than the conscious intention of the healer. Additional physical correlates to the healing using the Bengston method have included anomalous magnetic micropulsations in the surrounding space of the healing events, which may indicate a reduction in entropy when healing occurs. If this is the case, then perhaps it is information rather than energy that is being transmitted in the healing process, and if it is indeed information, that would be consistent with the apparent ability to store healing in material. The present study investigates whether stored and recorded healing intention can induce transcriptional changes in vitro. Success in this line of research would be a preliminary step to the development of a scalable delivery system for healing. Okay, let's go down to materials and methods. Okay, now again, I know I'm going to butcher this, but we're going to read a little bit about the cell cultures and exposure, and then we're going to skip down to I don't know. Let's take a look. Human breast carcinoma cells were cultured in a medium supplemented with 10% fetal bovine serum and maintained at 37 degrees Celsius in a humidified atmosphere with 5% CO2. So that's the cell culture. The murine memory tumor cells were cultured in Roswell Park Memorial Institute medium, supplemented with 10% fetal bovine serum in the same conditions that were used for the hands-on treatment. I'm not so sure about what all that means. I mean, that they're talking about what the cell culture is. Sterilized 4-ounce cotton rolls. This is exposure to energized cotton. And energized cotton is when they put their energy into the cotton. Sterilized 4-ounce cotton rolls were acquired from a local pharmacy, just regular cotton, and treated for 30 minutes by healers trained in the Bengston method. 100,000 cells were seated in 6 well plates. After 18 hours, a 4x6 inch piece of treated cotton was then placed on top of the 6 well plates for 24 hours. Cells were collected and processed for analysis, and for control, cells were not exposed to cotton. So, in other words, they treated some cotton for 30 minutes, and then after 18 hours, they placed the cotton on top of the plate, on top of the cells after 18 hours, and left it there for 24 hours. And then for a control, some of them were not exposed to the cotton. 3 people practiced the Bengston, recording of energy healing activity. 3 people practiced the Bengston healing method on cotton inside a solid steel, double-walled, electromagnetically shielded chamber for 5 minutes. As the treatment of cotton took place, magnetic and electromagnetic signals were recorded using 4 types of sensors. There's some magneto-resistant sensors manufactured by Honeywell, 2 antennas, some geomagnetometer, and custom PC software converted and saved the incoming sensor signals into the wave audio format. So what this is talking about is, up here they're talking about exposure to energized cotton. Here they're talking about 3 people recorded some type of something onto a wave file. And here it's going to talk about what they did with that wave file. Some flasks were placed in an incubator at 37 degrees Celsius for 5 minutes to 72 hours. 2 passive speakers were placed inside the incubator, and the speakers were Pyle Home from Brooklyn, New York. Just regular speakers were placed inside the incubator and connected in phase with speaker wires, yada yada, to an amplifier itself plugged into the speaker connector of a computer from which the recording was played repeatedly using Windows Media Player, just regular software. The volume of the computer and the player was at 50% setting. The volume of the amplifier was at 50%. Speakers were placed facing down on the top shelf, and the cells were placed on the lower shelf. So the speakers were on the top shelf, cells were on the lower shelf, directly underneath the speakers. In control settings, the same protocol was followed, except that no recording was played, using the ambient background noise as control condition. So what they did here, they took some cells, they put a speaker on both. On one set of cells, they played the sounds from the healing up here. On the other set of cells, they just played ambient background noise. Hands-on treatment of cancer cells. 750,000 cells were plated in some flasks and placed in a plastic chamber in the incubator for 18 hours before initiation of treatment. So some cells were placed in an incubator before the treatment. For the treatment, 3 healers practiced the Bengston method. 1 healer held the chamber while the 2 other performed the treatment remotely. Remotely means they were not there. During the whole time, the 3 healers were on a conference video call. Treatments took place in sessions of 1 hour. For the 2-hour treatment, cells were treated twice, 1 hour with a 30-minute break. For the 4-hour treatment, cells were treated in the same fashion on 2 consecutive days. Between the treatment, cells were placed back in the incubator. At the end of treatment, cells were collected in something, in Trizol, Thermo Fisher, Waltham, Massachusetts, and samples were processed as detailed below. So Trizol is a chemical solution used in the extraction of DNA, RNA, and proteins from cells. So right back here where it says at the end of treatment, cells were collected in Trizol, they were collected in a solution that is used in the extraction of DNA, RNA, and proteins from cells. Gene expression analysis by the QRT-PCR. And QRT-PCR means quantitative reverse transcription polymerase chain reaction. After exposure to treatment, treated cotton, recording, or hands-on, cells, and that was talked about up here. Here's hands-on, here is recording, and here is cotton. After exposure to treatment, cotton, recording, or hands-on, cells were collected in Trizol, that solution that we just spoke of, and total messenger RNA was extracted following the manufacturer's instructions. Complementary DNA was synthesized using RT-squared, maybe, first strand kit from 250 milligram ng of RNA expression. I'm not even going to read all this. This is how they, if you want to read on how they collected the cells. Here it is. 1 microliter of cDNA was used in the PCR reactions. Results were analyzed using this method right here. All right, I didn't get much out of that part, but I know they collected it in a certain fashion that most likely goes along with the scientific method. And you can read here for the statistical analysis. But what were the results? Effect of energized charged cotton on cell growth and cancer-related gene expression. Results from previous studies suggest that the Bengston method seems to exhibit anti-cancer activity. Cotton appears to be able to store the energy derived from this method and deliver these anti-cancer properties. Building on these observations and the previous work by Grad, we investigated whether charged cotton could induce measurable changes in cancer cells in vitro using MDA-MB231. And if you would like to know what this is, the MDA-MB231 cell line is an epithelial human breast cancer cell line that was established from a pleural effusion of a 51-year-old Caucasian female with a metastatic mammary adenocarcinoma 1 and is one of the most commonly used breast cancer cell lines in medical research laboratories. So we investigated whether charged cotton could induce measurable changes in the cancer cells in vitro using this line, carcinoma cells. Because of the mechanism of the therapeutic effect of the Bengston method is unknown, we initially screened for markers using the QRT-PCR assay comprising of 84 genes involved in cell growth and cancer. The list of genes is available in Supplemental Data S1. Cells were exposed to cotton treated by the Bengston method for 24 hours, and the control consisted of cells not exposed to cotton. Significant change in expression was found in 6 genes, suggesting the viability of the storage of healing information. While statistically significant, these changes in expression were all less than 2, suggesting a modest effect of cotton on transcriptional activity in MDA-MB231 cells. The next step focused on evaluation of a recording of the healing process. Here's Table 1. Significant change in 6 genes, talking about Table 1. I'm going to be honest, I don't even know what this is here, so I'm not going to read it if I don't even understand it. Maybe I'll understand it one day. That's why I'm doing the research here. Recordings of healing energy. In an attempt to capture the energy transferred when healers practice the Bengston method, a custom-made apparatus was designed at the Institute of Noetic Scientists. An apparatus was designed at the Institute of Noetic Sciences. In brief, it allowed the recordings of energy fields using multiple sensors while healers practiced the Bengston method to charge substances such as cotton. To isolate from surrounding contaminants, the system was set inside a Faraday chamber. Hang on. I want to show you what that is before we move on. See this lady inside the Faraday cage? Electricity can't get in. A Faraday cage or a Faraday shield or Faraday chamber is an enclosure used to block electromagnetic fields. Recorded data were converted in audio files, and spectral and temporal analysis were performed to identify potential variations between the control situation, empty chamber or chamber containing a neutral material, and an active situation, healers practicing the Bengston method inside the chamber. Some interesting short-term temporal effects were observed in some cases versus controls, and these may be the subject of further study. However, here we focus on the more consistently observed spectral changes, recording R18, which is right here, where 3 healers engaged in treating pieces of cotton showed the most consistent spectral differences versus the control. The control is down here, the other difference is up here. Spectral analysis covered the frequency range from below 0.1 Hz to about 20 kilohertz for recording R18. The only region where significant differences were observed was at frequencies below 20 Hz, and in particular below 5 Hz. For all 11 three-axis magnetometers were used to record the activity. One data channel on the y-axis indicated significantly elevated spectral content in the frequency range of 0.25 Hz to about 3 Hz. And you know, this right here, I've heard somebody else talk about this too over at the Monroe Institute, and that'll be another video, I guess. I think they talk about like 3 Hz to 8 Hz or something. I'm not sure, but we'll dive into that later. As compared to control recordings, the elevations in spectral levels between R18 and control results were typically 6 decibels or more, and in some cases 20 decibels or more. Figure 1, which is this, illustrates the spectral differences in the very low frequency range for 2 independent magnetometers. So 2 independent magnetometers came up with roughly the same result. The control here and a spectral elevation there. Okay. Recording R18 induces gene expression changes in breast cancer cells. Because R18 displayed a highly unusual spectral content, we then investigated whether this recording could produce biological activity. Of note, the spectral elevation is located in the range of very low frequencies, thus inaudible to humans. For this reason, the control used in this series of experiments was environmental background noise. So the control was background noise. We exposed MDA-MB231 cells to R18 and looked for gene expression changes by QRT-PCR. For the time of the exposure, cells were placed in an incubator containing 2 speakers plugged to a sound system described in Material and Methods. You can rewind to go back to Material and Methods. The control consisted of cells placed for the same amount of time in the incubator while the whole system was on but no recording was playing. Thus any potential interference from the speakers or the amplifier was present in both the control and experimental conditions. Initially, cells were exposed to R18 for 5 minutes, 30 minutes, 4 hours, 24 hours, 48 hours, or 72 hours. Expression of 84 genes involved in cancer pathways was screened using the same assay as for cells treated with cotton, as described above. Let's see. Previous studies suggested that there may be an active immune response induced by hands-on healing. Thus, we also screened 84 genes involved in the crosstalk between immunity and cancer. Of over 167 genes tested, exposure to R18 induced statistically significant changes, p being less than 0.05, in the expression of 68 genes when compared to the control for at least one time point tested. For 37 of these genes, the change was greater than 1.5-fold. Okay, so right here, this is, I'm not sure what this means. This interesting result suggests that the recording may exert an activity on cancer cells, though there was no clear pathway emerging from the genes affected. There were only 17 genes selected for further analysis in the 2 new independent experiments. And it goes on to talk about some of the, it starts talking about this, I'm not sure. Sometimes these become a little bit difficult to read, but you can go read it yourself here. We've discussed quite a bit of it here. We can't get everything. Okay, scroll down a bit. So all in all, this study represents the first steps of the development of an alternative delivery method of the Bengston healing method in order to be able to benefit from the therapeutic effect independent of the healers. The goal was to identify a vehicle for the healing that could be studied in laboratory settings and produce consistent changes in a living model compared to a control situation. We started these experimentations in vitro using cancer cells in a culture to screen by QRT-PCR changes occurring at the transcriptional level. The first vehicle that was investigated was cotton and has been clinically used to transfer the energy and reproduce healing properties previously. However, we did not identify strong transcriptional changes in breast cancer cells treated with cotton compared to control cells. So let me think about this one for a minute. However, we did not identify strong transcriptional changes in breast cancer cells treated with cotton compared to control cells. So I'm not sure. Then we assessed another method of storage by recording magnetic and electromagnetic signals originated from the healers practicing the Bengston method. This was done in collaboration with the Institute of Noetic Sciences, which engineered the sensors and conversion module to produce recording in an audible format, all inside a Faraday chamber. The recording of 3 healers charged cotton induced singular signals in low frequencies that were not observed in control situations, such as an empty chamber or the chamber containing untreated cotton. Previous studies have shown that low frequency signals or infrasound can increase sensitivity to chemotherapy and increase cell permeability. This recording, R18, was selected for further investigations. And for the purpose of screening, we used 2 QRT-PCR assays that include each 84 genes involved in cancer pathway. Let's see. Let's just keep scrolling down here a little bit. Okay, so there is little doubt but that some biological response has been simulated by both the healer and the recording of the healing energy. So some biological response has been stimulated by both, the consequences of that genomics effect and the careful evaluation of the several components to induce them require further study. Multiple questions need to be addressed, such as exposure time of subjects to energy recordings, the composition of the recording itself, what equipment can best capture the healer's input, and so on. Efforts to find answers to these questions are underway. In summary, the following suggestive conclusions may be drawn from the results, or from our results. 1. Reproducible biological changes have been induced by healing energy, whether by direct hands-on healing or using a recording of healing activity. Number 2, healing intention can be captured and released, thereby potentially allowing the phenomenon to be more widely disseminated. And number 3, hands-on delivery of the healing intention is stronger than with the recording used in this study, suggesting the possibility that the recording did not fully capture the healing potential. Okay. Some supplement. So, in this report, it's not great, but there are some things in here. They are right here. This is a summary. So there is evidence. Supplemental data. There's some acknowledgments. The authors thank David Dominic for both his support and suggestions. There's some footnotes. Let's see if we have anything else. Here are some references. I want to be studying about this guy right here, Grad. I've already got it up here. I'm going to be looking at him pretty soon. So go to the site, and I'm going to put these URLs in here, so you can look up the research yourself. Thank you. Have a great day.